Today we're going to start the nervous system, and let's begin with a short introduction. The nervous system and the endocrine system coordinate all the other organ systems to maintain homeostasis. The nervous system is fast, short-acting, where the endocrine system is slower but long-lasting. The nervous system is the most complex system in the body. The nervous system monitors the body's internal and external environments. It also integrates the sensory information. It coordinates the voluntary and involuntary responses. There are some divisions in the nervous system. The central nervous system is made up of the brain and the spinal cord. It integrates and coordinates input and output. The peripheral nervous system is all the neural tissue outside the central nervous system, and it's the connection between the central nervous system and the organs. There are two divisions, functional divisions that is, of the nervous system. The afferent division, this includes all the sensory receptors. These are the neurons that send information to the central nervous system. And the efferent division, these are neurons that send information from the central nervous system to the effectors. Effectors are either muscles or glands. It can be further divided into the somatic nervous system, the SNS, and that controls skeletal muscle, or the autonomic nervous system, and that controls the smooth, cardiac, and the glands. The smooth muscle, the cardiac muscle, and the glands. That can be the autonomic nervous system, the ANS, can be further divided into the sympathetic division or the parasympathetic division. Parasympathetic is more relaxation, where the sympathetic division is fight or flight. This concept map will explain the overall process. Let's start with number one. The sensory receptors, the five senses, and the internal sensory receptors are part of the afferent division, and they're receiving the information and bringing it to the central nervous system, which is the brain and spinal cord. At the brain and spinal cord, they process the information, and then they send a command out from the central nervous system on the efferent division. And it will either go to the somatic system or the autonomic system, or both. And if it goes to the somatic system, it will go to the skeletal muscles. In the autonomic nervous system, it'll either go the parasympathetic division or the sympathetic division. Let's go on to section 8.2. Let's go into the actual anatomy of the nervous system. Let's begin with the nerve cells. They're called neurons, and neurons are cells that communicate with one another. They have a basic structure. They have a cell body. They have dendrites. Dendrites receive signals. Axons, and they carry signals away from the cell body to the next cell. And axonal terminals, they're bulb-shaped endings that form a synapse with the next cell. Neurons have very limited ability to regenerate. So when they're damaged or destroyed, usually their functionality is lost. This is a picture of the neuron. Make sure you know this. This will be on your test. Know the dendrites, the cell body, know the nucleus, the mitochondria, the Golgi apparatus. Make sure you know the axon and the axonal terminals. There are three types of neurons. There are multipolar neurons. They're the most common in the central nervous system, and they have two or more dendrites and one axon. Unipolar neurons, and they have the cell body off to one side. And then bipolar neurons have one dendrite and one axon, and the cell body in the middle. They're very rare. This is a picture of the three types of neurons. Sensory neurons are also called afferent neurons. They detect stimuli concerning the outside world in the form of external receptors and are positioned in it in the form of proprioceptors. And then they also are visceral or internal receptors, and they monitor our internal organs. Motor neurons are called efferent neurons, and they're either somatic motor neurons or they actually move the skeletal muscle, or visceral motor neurons where they innervate the cardiac muscle, the smooth muscle, and the glands. Lastly are the interneurons, and they're the neurons in the central nervous system, the brain and spinal cord. There's over 20 billion. They're the most numerous, and they have the higher functions such as memory, planning, and learning. Now, there are 6 basic types of cells that are supportive cells that make up the nervous system. What they do is they support and help the neurons to function throughout a human lifetime. There are 4 types found in the central nervous system, and there's 2 types in the peripheral nervous system. In the central nervous system, we have astrocytes, oligodendrocytes, microglia, and ependymal cells. In the peripheral nervous system, we have satellite cells and Schwann cells. Astrocytes are the most numerous neuroglia, and they maintain the blood-brain barrier in the central nervous system. Oligodendrocytes are involved in insulating membranous wrapping around axons called myelin. In other words, it's the white matter of the central nervous system, and they're myelinated axons. Cell bodies help make up the gray matter. However, there are some axons that are unmyelinated. The microglia are the phagocytotic cells derived from white blood cells, and they engulf pathogens and cellular waste. Finally, the ependymal cells, they line the fluid-filled central canal of the spinal cord and the ventricles of the brain. They're also involved in producing and circulating the cerebral spinal fluid, the CSF, around the central nervous system. This is an image showing you the white and gray matter. Now, the neuroglial cells in the peripheral nervous system, the satellite cells, are the supportive cells, and they're similar or parallel to the astrocytes, but they're found in the peripheral nervous system. And the Schwann cells cover every axon in the peripheral nervous system. And what they do is they produce myelin. Again, this shows you the Schwann cell and the myelin covering. Now, you need to know a couple things. We have 2 major sets of nerves called spinal nerves and cranial nerves. They both have sensory and motor components. A couple things that you do need to know. There's gray matter and white matter. This kind of shows you in the peripheral nervous system, the gray matter is in the ganglia and the neural cell bodies, where the white matter is made up of the myelinated axons. In the central nervous system, the gray matter is made of the cell bodies and the glial cells, whereas the white matter is again made of the myelinated axons. Now let's actually talk about membrane potential. Most cells have what we call a resting membrane potential. That means there's a charge involved. And an undisturbed cell has a resting membrane potential measured inside the cell in millivolts. The resting membrane potential of neurons is negative 70 millivolts. There are some factors that help determine that. We have an extracellular fluid that's high in sodium and chlorine. We have an intracellular fluid high in potassium and proteins. There are some channels that are always open, and they're called leak channels. And some are open or closed, and they're called gated channels. This is some of the anatomy. Sodium can leak in, and that allows potassium to leak out faster. We have a sodium-potassium exchange pump, and that exchanges 3 sodium for every 2 potassium. And this helps maintain the resting membrane potential at negative 70 millivolts. This shows you the actual cell membrane and the extracellular and intracellular fluid. Now, some things will change the membrane potential. It can include cellular exposure to chemicals, mechanical pressure, temperature, changes in the extracellular ion concentration, and that can result in the opening of a gated channel. Now, when gated channels open, some things happen. The opening of the sodium channels will result in sodium rushing in. It's called depolarization. Opening of the potassium channels result in potassium rushing out. That's called hyperpolarization. And of course, that will then have to return to resting state, and that's called repolarization. Now, in order for a nerve to actually have an impulse, it has to reach what's called critical point. So, a change in the membrane that travels the entire length of the neuron is called a nerve impulse. In order to do that, it has to reach a critical point, and it's called threshold. Then an action potential will occur. All action potentials are all or none. So a membrane can't respond unless it hits that threshold level. At that point, an action potential will happen. The membrane cannot respond until for further stimulation until the refractory period is over. So here's what you need to know. We need to reach threshold level, which is negative 60 millivolts. At threshold, it's all or none, and when that happens, the sodium channels will open, the sodium will move in, and then when we reach step 3, the sodium channels will close and the potassium channels will open. At that point, the potassium will rush out. Now repolarization begins. At step 4, the potassium channels will close and both the sodium and potassium channels will return to resting state. A refractory period is from the point of threshold until repolarization happens. At that point, the membrane cannot respond to further stimulation. Now, there's two types of propagation, or the movement of the membrane, movement of the action potential down the membrane. One is called continuous propagation, and that occurs in the unmyelinated fibers and is relatively slow. And then there's saltatory propagation, which is in myelinated axons, and it's much faster.